Assalamu alaikum, everyone, and I hope everyone is doing well and all of you have received your O levels grades. So, congratulations on officially completing your O levels and now beginning officially your journey in A levels. So, good luck with your A levels and good luck with this academic year once again. So, we briefly started the chapter of cell membrane in the previous class. And now I'm thinking to go over it briefly. We'll go over the previous concept and then we'll begin, inshallah. All right. So the reason why we're starting with chapter number 4 instead of chapter number 2, it is because chapter 4 is slightly easier than chapter 2 and 3. And in order to connect with chapter 1 and in order to make a strong connection with chapter 1, we need to do chapter 4 first. This is exactly how I did in my A levels. My teacher taught me chapter 1 and then she taught us chapter 4, because usually when we'll go over the past papers during our live class, we'll see that many of the questions are interconnected with chapter 1 and chapter 4. Okay, so in order to make a proper connection between chapter 1 and 4, when we solve the past papers, it will be easier for you to understand the pattern of the questions. All right, so let's begin, inshallah. We discussed that now you won't be calling cell membrane as cell membrane. You'll now call it as cell surface membrane, plasma membrane, or phospholipid bilayer. All right. So starting with the structure of cell membrane, cell surface membrane, or plasma membrane, or phospholipid bilayer, first you need to remember that plasma membrane is of 7 nanometers. We discussed about this size in chapter 1 as well. And the reason we discussed it was that light microscope has a resolution of 200 nanometers, and it is nearly impossible for us to see cell surface membrane in light microscope due to its small size, which is 7 nm. So we can only see plasma membrane in electron microscope because the resolution of electron microscope is far more better than light microscope. The resolution of electron microscope is 0.5 nanometers. Just a quick reminder. All right, so let's begin with the shape. This was the size, 7 nanometers is the size. Now we are going through the shape of plasma membrane. All right, so basically we studied that cell membrane somehow looks like this, but it looks like this. This is the actual structure of cell surface membrane, where we have few components that make up the cell surface membrane, and those are, starting off with this first a circular head that we can see. This is known as phosphate head. All right, so you can see this word written over here, phospholipid. So I'm breaking this word into two. I'm saying that this circular part is my phosphate head. All these circular structures, these are made up of phosphate. So this is why we are saying that these are phosphate heads. All right. So phospholipid has first part of phosphate head, and beneath these phosphate head, you can see few tails projecting out. So each circular head has two fatty acid tails that are projecting out of it. These are made up of lipid. All right. So this lipid portion, now we'll call this fatty acid tails. What are these? These are fatty, F means fatty, acid tails. All right, so phospholipid means that this component, I'm just talking about this component, all right? I'm talking about this component. So this component, this has a phosphate head, the circular part is phosphate head, and these tails are fatty acid tails. So we call this structure as phospholipid. Now, moving towards the characteristic of phosphate head, it is hydrophilic. So hydro means water and philic means loving. So if we break hydro, this means water again. So try to break these words and try to understand their meaning, and then it will be easier for you to remember, because there's a lot of detail that will be coming up in the chapters that we have to cover. So there's a lot of detail. So it's better to break these words and to understand their simpler meaning. Okay, so philic means loving. So in actual, what this means is that phosphate head is water loving, or you will call it hydrophilic, and it is polar. So we also discussed why it is polar, because it has charges. Okay. Then these fatty acetals, these are hydrophobic. Again, if you break this word hydro, it means water, and phobia for, because you see we all have certain phobias. So phobia, from that word phobia, it means pairing. So fatty acetals are water pairing, and these are non-polar in nature. Next component that we have in plasma membrane is protein. So we have basically two types of protein. We can see over here. This is also a protein. This is also a protein. This also is a protein. And so basically what you need to remember is that there are two types of proteins: integral proteins or intrinsic proteins, or peripheral proteins. We'll talk deeply about channel proteins and carrier proteins in the upcoming slides. But for now, try to remember that there are two types of protein in plasma membrane: integral and peripheral protein. The other name for integral protein is intrinsic protein, and the other name for peripheral protein is extrinsic protein. You can try to make notes as well. When I'm telling something important, make small notes, because sometimes in your paper there might be another word and you may get confused. So in order to remove those confusions, just write integral protein / intrinsic protein. We'll talk deeply about channel and carrier proteins in the upcoming slides. So let's just ignore it. Try to remember its shape for now. Then we have glycoproteins and glycolipids. There's just glycoprotein, but glycolipids look exactly the same. Okay, so glycoproteins, we'll talk about this as well, that these are certain projections which, okay, so glyco means carbohydrate, obviously, and protein, the protein, it's a biological molecule. So glycoprotein is a biological molecule made up of carbohydrate and protein. And these are certain chain-like projections that you can see. They are propagating outwards towards, okay. So if I call this extracellular, I hope you'll be able to understand that extracellular is the region which is outside of the cell. And this should be intracellular. Intracellular means inside the cell. So this is intracellular and this is extracellular. EC means extracellular. So glycoprotein chains are projecting outwards. Where are they projecting? They're projecting towards extracellular component. All right. So we'll talk extensively about the functions, about the roles that all of these components are playing. All right. So then you also need to remember that in this plasma membrane, you will see cholesterol in middle many times, and there's an important function of cholesterol as well. So we'll talk about it deeply as well. I hope we are clear with the structure. And you also need to remember one more thing, that this question comes about, I think I have seen this question almost in all of the past papers. So there's this 4-mark question, and they say describe fluid mosaic model. You can see this word, fluid mosaic model. So whatever I have explained up till now, this is the entire fluid mosaic model. We'll discuss what fluid mosaic means, but try to remember that whatever structure that we have covered of plasma membrane right now, it is exactly what you need to write in your script when you'll answer the questions for fluid mosaic model. This model is basically telling you the structure and the components that make up the entire plasma membrane. All right, so this is a 4-mark question which repeatedly comes in your past papers. Now, again, so this is, okay, so the name of this plasma membrane is phospholipid bilayer. All right, so we now know that phospholipid, what is the meaning of phospholipid, right? We know that there's a phosphate head, these are the fatty acid tails, but you need to understand the meaning of bilayer. Bi means two, right? So basically it is trying to tell us that the plasma membrane has a bilayer. Bi means two, and layer, we can see that this is the first layer. This is the first layer, and this is the first layer, then this is the second layer. So this is how the cell surface membrane is arranged. It is arranged in a bilayer manner. All right. Now moving towards, and this, you can see this is an intrinsic protein, and this is a peripheral protein which is projecting outwards into this extracellular component. This is also an extrinsic protein, but this is an intrinsic protein which is inside the phospholipid bilayer. All right, so now moving towards the detailed part. Now, phospholipid bilayer consists of phosphate head, which is hydrophilic, we already talked about it, and fatty acid tails, which are hydrophobic, which are water-hating in nature. We already discussed this part, right? Now, fluid mosaic model refers to the movement of phospholipids, while mosaic refers to the scattered protein and glycoprotein in the phospholipid bilayer. So I just mentioned that this is a question which will need 4 pointers, right? So you need, this is just the first pointer. This is just explaining that fluid means move, fluid means water, like, so water means, water indicates something with the movement part, right? So phospholipid basically is trying to tell you that a phospholipid is in continuous motion due to fluidity. So there is some sort of fluidity in the entire membrane. It is not static, it is not in a frozen manner. So it is continuously moving due to fluidity. So there are certain factors that are influencing the fluidity, and why is there fluidity in the cell surface membrane. We'll talk about those factors as well. But for now, try to understand that plasma membrane is not static. It is not in a frozen manner. It is capable to move. So, and there are many factors that influence the movement of this entire membrane. So there is some sort of fluidity, and fluid means that there is some movement of phospholipid, while mosaic means that there are scattered protein. So mark this word: scattered protein is the most um it is the most significant keyword of your script. You'll often find scattered protein in your marking schemes as well. So this is a keyword that will give you 1 mark. All right, and this movement of phospholipid, this is the another keyword that should be a part of your script while you answer fluid mosaic model. So side by side, I'll also let you know the keywords that should be in your script. ठीक है? Because these words, you'll see them in your marking scheme as well. So we need to understand the art of writing those keywords that will help us achieve marks. Okay? Because currently there are many students that they were not my students, but many relatives of mine, they were calling me and they were saying that we wrote everything in our papers, but we did not get those grades that they were expecting. So this is my conclusion that um they're they're certain that they have written the best scripts, but I'm pretty much sure that they might have missed these keywords, and CIE requires that you write those keywords. Only then they'll give you marks. If for example the question was about Fajr and you are writing about Zohar, you're giving the details of Zohar prayer while the question was asking you about Fajr prayer. This is just an example, okay? So why are you giving the details of Zohar prayer when the question has asked you about Fajr prayer, right? You need to write it, how many farais are in Fajr while you are writing the farais of Zohar. So why will a CI examiner give you marks? Okay, so just try to understand the point that you need to be specific with your keywords. Only then you'll get marks. Only then you'll get your grade. So instead of complaining that we wrote everything in our paper, yet we did not receive our grades, so we need to recheck ourselves that what exactly did we write in our paper. And this usually happens when we do not practice past papers enough. So inshallah, we'll practice past papers. We already practiced the past papers of cells. We'll also discuss the past papers of cell membrane, and then we'll do yearlies once we finish our syllabus. So I'm just emphasizing upon keywords. I'm emphasizing on making notes. I'm emphasizing on what else am I emphasizing on? I'm also emphasizing on homeworks and tests. I haven't posted any homework as yet, but I will. And I'll also take a test of cells and cell surface membrane combined in, I think, in the next week or somewhere around it. So try to be sincere with your studies and try to be sincere with whatever you are doing. You know where you stand after your O-levels results. And I'm not demotivating anyone. I'm just saying that be sincere, and only then you'll be able to get grades that you deserve. And only then you'll be able to succeed, inshallah. So yeah, let's come back. And I was just saying that mark these keywords. These are highly important and they should be a part of your script even when I take your paper, when I take your exam, okay? Okay. So fluid mosaic refers to the movement of phospholipid. I mentioned that it is fluid in nature, while there are scattered proteins. So there are two types of protein in plasma membrane, extrinsic and intrinsic, and they are scattered over the membrane. All right, and there's one more thing that influences the mosaic model, which is glycoproteins and glycolipids. So those chain-like structures also give the appearance of mosaic, the glycoproteins and glycolipids. All right. Then we have phospholipid bilayer. We already talked about the two types of protein, intrinsic and extrinsic. And we also discussed that it has glycolipids and glycoproteins. So these are two biological molecules, carbohydrates and lipids, carbohydrates and proteins. So these are chain-like projections that are propagating outwards in the extracellular component. Now let's talk about intrinsic protein. So intrinsic proteins are those proteins which are inside your membrane. You can also call them transmembrane proteins, and they span over the entire membrane. So there are certain portions that are exposed on the extracellular side and cytoplasmic sides, and transmembrane proteins, they function as channels and carriers and receptor proteins. So basically, when we were looking at this picture, you can see that there are two more types of protein, channel protein and carrier protein. So basically, these are your integral proteins or intrinsic proteins, and they play a very important role. So carrier protein plays an important role in active transport, while channel protein plays an active role in facilitated diffusion. We'll talk what facilitated diffusion it is. It is slightly different from the simple diffusions, just a slight difference. So as for now, remember that integral proteins are of two types. There's furthermore categorization of integral proteins or intrinsic proteins, which is channel protein and carrier protein. Okay. And yeah. So here's a more description. Channel proteins form pores through which specific ions or molecules can pass, allowing for facilitated diffusion across the membrane. Then, okay, so let me explain this. For example, if this is your channel protein, then what they're trying to say is that certain ions and certain molecules can pass through this channel. So these channel proteins provide a pathway for ions and molecules to pass through it, and then they can go inside the cell. Okay. So basically, they're just trying to say that channel protein provides a pathway for ions and molecules, and this is usually essential in facilitative diffusion. Then we have the other category of integral proteins, which is carrier proteins, and carrier proteins play an active role in active transport. Okay, so these bind to specific molecules on one side of the membrane, undergo a conformational change, and transport the molecule to the other side. So carrier proteins are slightly different from channel protein because channel proteins were providing a pathway, while carrier proteins somewhat, for example, if they look like this, they do not look like this. I'm just trying to explain you that if they look like this. So you can see that there is a junction, there's a depression. On this depression, a molecule will come and bind and will induce a conformational change in this carrier protein. And what will happen that, for example, now you can see that this carrier protein is projecting outwards, right? When this molecule will induce a conformational change, it will bend towards the other side. For example, like this, it will bend towards the other side and will allow the molecule to move inside the cell. Okay, so this is what all of this English means, that through this conformational change, the carrier protein swaps its position, it goes upside down, and the molecule is then able to propagate inside. Okay. Then we have receptor proteins. Receptor protein is also a type of intrinsic proteins, and this concept of signaling, I'm not discussing it right now because we'll deeply discuss signaling in the upcoming slides as well. We have this whole subtopic of cell signaling. Slightly technical, but let's discuss it over here as well. Receptor proteins, these proteins bind to signaling molecules, which are ligands outside the cell, and these transmit signals inside the cell, initiating a cellular response. So, okay, let's discuss this part when cell signaling comes, okay? Because this is slightly technical. We'll come back to it after doing a few slides. Okay. let's discuss periperal proteins. These are your extrinsic proteins as well. So they are not embedded into the lipid bilayer. They are over the surface of cell membrane. They are not inside there. They are on the surface of the cell membrane. And instead, they are loosely attached to the membrane surface, often through interactions with integral proteins or by associating with the polar head groups of phospholipids. So basically, they're just trying to tell you the position of extrinsic proteins, that they are not deeply embedded into the inside of the cell membrane. Rather, they are loosely, they are placed over the membrane. Okay, now here is the concept of cell signaling. So some peripheral proteins are involved in signal transduction pathways. So signal transduction literally means, okay, by the way, this is also the definition for cell signaling. If it comes for like 2 marks, you can write this definition for cell signaling. Okay, so cell signaling basically means that sometimes there are many types of signals that are coming into the cells. It could be anything. It could be an hormone, it could be an enzyme, it could be a neurotransmitter that is coming towards the cell. So basically what happens is, for example, if this is a cell, okay, if this is a cell and it has a receptor protein. So for example, an hormone is coming, or a neurotransmitter is coming, or an enzyme is coming. So this is a signal. Okay, this signal is coming towards the receptor protein. Receptor protein is going to accept this signal, and this signal may have certain message, obviously, or else why would it move towards a cell? So, for example, if this is a neurotransmitter and it is coming towards the cell, so it will bind with this receptor protein, and this will cause some conformational change within the receptor, and then whatever the message that this neurotransmitter is carrying, it will move inside the cell, and what do we say, some signal or some message will be transmitted inside the cell. So this is just a brief overview of cell signaling. These are not the complete steps, because we were discussing peripheral proteins, that peripheral proteins are involved in cell signaling, and I'm just trying to explain that cell signaling is a process in which certain molecules are coming with a message, and they bind with the receptor proteins, they induce a conformational change, and then that signal propagates inside the cell, and whatever message it has, it is executed within the cell. Okay. Then peripheral. So this is peripheral proteins function. This is the first function, and now we are discussing the other function, which is enzymatic activity. So some peripheral proteins act as enzymes that catalyze reactions on the membrane surface. So basically, you need to remember all the functions of proteins. You need to remember the functions of peripheral proteins. You need to remember the functions of intrinsic proteins as well. Okay, that they act as channel protein, carrier protein, receptor protein, while peripheral proteins act as cell signaling molecules, and they also play a role in enzymatic activities where they themselves act as enzymes and catalyze reactions. Okay. Next. So, okay, now moving towards this. Fluidity. We said that plasma membrane is fluid in nature. So there are certain factors that affect the fluidity of membrane, and those factors are: number one. So up till now, you should have this concept that cell membrane is not static, it is fluid, and it is continuously in a moving manner. And what are those factors that influence the fluidity of membrane? We are discussing those now. So first factor is tail length. So also these are very important. They can also come up for 4 marks, and they do come for 4 marks as well. Okay, so try to understand this. Tail length. We said that phosphate has certain tails. Like each phosphate has two fatty acid tails, right? So this is the first factor which influences the fluidity of membrane, which is tail length. So it says that longer the tail, lower the fluidity of the membrane. So if the tail is longer, then the membrane will become less fluid. And you need to write this word less fluid or lower fluidity. You do not have to write decreased fluidity or increased fluidity. Okay? Write the proper word lower fluidity or lesser the fluidity. So if this fatty acid tail is longer, then the fluidity will be lesser. And if these tails are shorter, then fluidity will increase. Then saturation of fatty acid tail. Okay, So before discussing this, I want to explain you the concept of saturation and unsaturation for those who do not remember O levels chemistry's organic part. Okay, I'm sure you guys did organic chemistry in O levels, but I'm sure that you do not remember it anymore. So basically, you might have seen that sometimes there are double bonds within two carbons, and sometimes there is a single bond. So this is a single bond, while this is a double bond. Okay. So whenever we have double bonds, we call this as unsaturated. Okay. This is unsaturated. And why we call this unsaturated? It is because any molecule, for example, a bromine might come and it may break these double bonds. And when these double bonds are broken, bromine can attach with carbon. Okay. So this is a slightly, we went into chemistry. Let's come back to biology. And when we have a single bond, this is a saturated molecule. Okay. So just try to remember this, that when we have single bond, that molecule is saturated, and when we have double bonds, that molecule is unsaturated. Okay, so saturation of fatty acetate. A higher degree of unsaturation in the tail leads to increased membrane fluidity. So whenever we have unsaturated molecules, fluidity of the membrane will increase. So the more unsaturated the tail is, the more fluid the membrane will be. Okay, so unsaturated molecules will favor the fluidity of membrane, while saturated molecules will not favor the fluidity. Okay. Now, at low temperatures, cholesterol increases the fluidity of membrane. At low temperatures, now here comes the function of cholesterol as well. So if your question asks you about the functions of cholesterol, you can write this. Okay. But this is also one of the factors which is influencing the fluidity. At low temperatures, cholesterol increases the fluidity of the membrane, preventing it from being too rigid. This is because it prevents close packing of phospholipid tails, and at high temperature, cholesterol decreases the fluidity of the membrane and stabilizes the cell. Okay, so this is a very easy concept to understand, that when there is a condition of low temperature, or when there is cold enough, then cholesterol will increase the fluidity of the membrane, so the membrane does not become static or it does not become rigid, because as we said, that membrane is in continuous fluidity. Okay, so to maintain fluidity, cholesterol at low temperatures will increase the fluidity. Okay, while an inverse will happen when a condition of high temperature is there. So these are basically four factors which are influencing the fluidity of membrane. Now, moving towards glycoproteins and glycolipids. So glycoproteins play a critical role in identifying cells. So now we are doing the functions of glycoprotein and glycolipids. Again, this might come for 4 marks again. Glycoproteins play a critical role in identifying cells, which is important for immune responses. They help the immune system distinguish between cells and foreign cells, such as pathogens. So this is a concept. This is a concept of immunity, and you do have this chapter of immunity, and I think it's probably the 10th chapter. Yeah. So basically, what happens is that these projections, they're acting as markers. So they differentiate between cell to cell, and they identify that how cells are different and how certain cells are important in immune responses. So these junctions, these chain-like structures, they are playing an important role in immune responses, as they act like markers. And for example, okay, so they may act like highlighters, because they are trying to identify the cells. They are trying to distinguish between the cells that are of body and the cells that are foreign in nature. Foreign cells could be of pathogens, and pathogens are basically the microorganisms. They can be viruses, they can be bacteria. Okay, so basically what happens is they act like highlighters. They highlight those cells that are foreign, so an immune response can occur. Okay, so this is the first function of glycoproteins or glycolipids. Then glycoproteins act as receptors for hormones, neurotransmitters, and other signaling molecules. When these molecules bind to glycoproteins, they trigger a response within the cell, leading to various cellular activities. So basically, like peripheral proteins, they also act as receptors for signaling molecules. And what were signaling molecules? They carry some sort of signals within themselves. They could be hormones, they could be neurotransmitters. And when they bind with those receptor proteins, they induce a conformational change, and then a transduction of signal would take place. So this is the second function. The third function is that glycoproteins help cells adhere. Adhere means to stick. They help the cells to stick to each other, which is vital for the formation of tissues and for maintaining the structures of multicellular organisms. This is also an important role of glycoproteins or glycolipids. That these projections that are in extracellular fluid, they are act like a glue and they bind the cells with one another. And this is exactly how our tissues are made, by the sticking of cells. Then number 4, glycoproteins contribute to the stability and flexibility of cell membrane by interacting with other membrane components. Okay, so these are the functions of glycoprotein and glycolipids. Now coming towards cell signaling. So cell signaling, cell produce and release signaling molecules, and these signaling molecules are known as ligands. So ligands could be hormones, neurotransmitter, or other signaling molecules. And why are these signaling molecules released? Because they carry some sort of message for cells. Then ligands are signaling molecules. Okay? Ligands are often secreted from cells via exocytosis. Do not worry about exocytosis. We are going to discuss it in the upcoming slides. Okay? Just remember this word exocytosis, where vesicles containing the ligands fuse with the cell membrane and release their contents into the extracellular space. Ligands can diffuse through the extracellular fluid to reach nearby target cells in the case of local signaling. And in the case of endocrine signaling, ligands such as hormones are transported through the bloodstream or other bodily fluids to reach distant target cells. So there's a slight difference. There's a slight difference between local signaling and endocrine signaling, that when there's this case of local signaling, ligands diffuse into the extracellular fluid and they reach the nearby target cells as soon as possible. Okay, we all just discussed what extracellular fluid is. Then, for example, if a hormone is released, so hormones have to travel, right? So when hormones travel through the bloodstreams, they take certain time. And basically, when hormones are released, they are released just to reach the distant target cells. Okay, so local signaling happens in a faster manner as compared to endocrine signaling. Okay, so ligands bind specifically to receptor proteins. We discussed it on the surface of target cells. Okay. These receptors are usually highly specific to particular ligands. Obviously, just try to recall the concept of enzyme and substrate. That enzyme used to be of certain shape, right? And the substrate would be of the complementary shape as well. So this is exactly the case with ligands and cell signaling, that the ligands, for example, if a ligand is circular in shape, then for sure the receptor is going to be of circular shape as well, so that the signaling molecule could be accepted within the receptor proteins. Okay. Yeah. So then they bind, obviously they're going to bind, and then that receptor protein undergoes a conformational change, which initiates the signaling cascade inside the target cell. And this process can activate various intracellular pathways leading to specific cellular response. So whatever that message is, it basically, when that conformational change has happened, a signaling cascade will happen. A signaling cascade means that many messages are going to be initiated, that what a cell particularly has to do with that signaling molecule, and then finally that response is triggered, which was brought originally by that hormone or by that neurotransmitter. Then the activated receptor typically triggers a series of intracellular events. So this is an important keyword. Often involving secondary messengers, protein kinases, this is just an example, or other signaling molecules, which amplify the signal and direct it towards cellular responses. Okay. Okay, so now let's come towards the movement. There are 5 types of movements that we are going to discuss, that how ions and molecules move. So first one is diffusion. Diffusion, we have already studied this in our O level, that diffusion is the movement of ions and molecules from a region of higher concentration to a region of lower concentration down a gradient, and this is usually a passive process which does not require energy. So these are certain factors which influence diffusion, that the if the concentration gradient is steeper, then the rate of diffusion will also increase, which means that, for example, if um, these are certain molecules, trying to make molecules, you can ignore my drawing because this is not an arts class. I'm just trying to explain you a few concepts. For example, these are molecules, okay? And this is the other part, this is the other region where these molecules have to migrate. So obviously we can see that there is more steepness. So this steepness will increase the rate of diffusion, and molecules are going to travel as soon as possible. Number two, as the temperature rises, the diffusion rate accelerates, and as there is an increase in surface area, it will also increase the rate of diffusion. I'm just skimming it over because we have already studied the entire concept of diffusion in O levels. And as the distance over which diffusion must occur increases, the diffusion rate will decrease. So this is very obvious that if, for example, the distance for diffusion is more, then diffusion will occur at a decreased rate. Then again, molecules like fat, which are small and non-polar, diffuse more easily across the cell membrane because they are soluble in phospholipid tails. Okay. Now coming towards facilitated diffusion. So this is something new. Facilitated diffusion is a type of passive transport that allows specific molecules or ions to move across the cell membrane with the help of membrane proteins. So we discussed that facilitated diffusion requires channel proteins. So we have two types of membrane protein, channel and carrier. So basically in facilitated diffusion, only specific molecules can move across the cell, and they require channel protein for sure, because in diffusion, simple diffusion, we do not need channel proteins, or we do not need carrier proteins, and it usually allows all the molecules, all the small molecules, to pass across the cell membrane. But as far as facilitated diffusion is concerned, it also allows polar molecules. It also allows slightly larger molecules to pass through the channel protein, and this is why the name is facilitated diffusion. Okay. Then, unlike simple diffusion, which occurs directly through the lipid bilayer, facilitated diffusion relies on specialized proteins to transport substances that cannot easily pass through the hydrophobic core of the membrane. So remember that there are many fatty acid tails. So those fatty acid tails, when they are in a bilayer, they are in a bilayer manner, it forms a hydrophobic core. So hydrophobic core basically does not allow polar molecules to pass through. So this is why facilitated diffusion is there to to facilitate the process of diffusion of those molecules which cannot pass through simple diffusion. And just remember this thing, that transport proteins are involved in facilitated diffusion, and they allow certain molecules or ion to pass through, and this process does not require energy. And this is the example, that glucose molecules, that they are too large and they're polar to diffuse directly through the lipid bilayer. They're transported into cells using facilitated diffusion. So basically, facilitated diffusion allows specific molecules, such as they could be large and they could be polar. So large and polar molecules are diffused through lipid bilayer only with the help of facilitated diffusion, because there is a separate channel for those molecules to pass. All right. Then we have osmosis. So osmosis is the net movement of water molecules from a region of higher water potential to a region of lower water potential through a partially permeable membrane as a result of the random motion. We have already covered this in O levels, but this is a slightly technical part that we need to know this word, the meaning of this word rather, water potential. It is the tendency of water to move out of a solution. Okay. You will not be asked on these things, but you need to know, because these are certain concepts which are associated with osmosis. So water potential is the ability of water molecules to move out of a solution, and the movement of water always moves down its water potential gradient. Similarly, it is the similar case of diffusion rather, but it just involves water molecules. And water potential is represented by the Greek letter. This is important. This might come in your paper once. I haven't seen a question, but this might come. Okay, so remember that water potential has a Greek letter and this is how it looks. Then effect of solute concentration. So this is very basic thing to understand, that water potential will become negative when the solute concentration is high. So for example, if in a solution you have 10% of salt and 5% of water, so obviously your water potential is going to be negative because you have more solute rather than water. Okay, so just remember this. Now, this is just a quick recap from your O levels, that isotonic is the solution where water potential is equal. So if you place plant cell in an isotonic solution, nothing will happen. Nothing should happen to a plant cell when it's placed in an isotonic solution. It should not be placid. Nothing will happen to, there will be no change into the shape of plant cell when it's placed in isotonic solution. But when it's placed in hypotonic solution, hypotonic is a solution where water potential is very high and water molecules are going to move from the outside of the solution to the inside of plant cell, and it will result in swelling of the plant cell. In the swelling part of the plant cell is known as turgid. So when we place a plant cell in a high water potential or in a hypotonic solution, this plant cell will become turgid. Then when we place plant in a solution which has low water potential or it's hypertonic, then it will become plasmolized because the water will move out of this plant cell. Okay. Okay. So this is the same case with red blood cells, but we do not say that when we are placing a animal cell in a hypotonic, isotonic, or hypertonic solution. Rather, we say that in this scenario, red blood cell was placed in a solution of solution of high water potential because the cell has eventually got burst. And why it got burst? Because it does not have a cell wall to support its structure. So as the water moved inside, the cell swelled and then it burst. In a normal condition, when water potentials are equal, nothing will happen. And if the red blood cell is placed in a solution of low water potential, then water will move outside and eventually the cell will shrink. Now moving towards active transport. So active transport, it is the movement of molecules or ions through transport proteins. And now you should remember this transport proteins here is carrier protein. For facilitated diffusion, channel proteins play their part, and for active transport, carrier proteins are going to play their part. And this movement happens usually against the concentration gradient using energy from ATP. So this process involves ATP. This process is facilitated by specific carrier, usually by carrier proteins and sometimes with channel proteins, but we only mention carrier proteins, okay, that are specialized for the type of molecule they transport. And the example of active transport is sodium-potassium pump, which pumps 3 sodium ions out of the cell and 2 potassium ions inside the cell against their concentration gradient. Then we have bulk transport. So this is a whole new movement process for you. Bulk transport is of 2 types, and as far as we have discussed diffusion, facilitated diffusion, osmosis, and active transport, we were just talking about ions and molecules. But what if we have a large quantity of substances that we need to transport? It could be antibodies, it could be anything, right? So what happens? We were only talking about ions and molecules. Now, if we have something very big, then we need bulk transport. Okay? So in bulk transport, what happens is that it is basically a vesicular transport. So basically in bulk transport, things are transported in form of vesicles. And it usually involves large quantities of substances which move into or out of the cells. So this process requires energy like active transport. All the other processes did not require energy. Okay, so remember this, that facilitated diffusion, simple diffusion, facilitated diffusion, osmosis, they do not require energy, while active transport and bulk transport, they require energy because they involve movement of large volume of materials and the alteration of cell membrane structure. So basically, when we talk about bulk transport, we need to know that a bulk transport is of two types. Number one, endocytosis, and number two, exocytosis. So endocytosis basically means, endo means inside. So endocytosis means that this is a process by which cells internalize large molecules, particles, or liquid from their external environment. And endocytosis is of further two types, phagocytosis and pinocytosis. So there's just a minor difference between both. So remember this, that in endocytosis, large substances are moving inside the cell. Okay. And endocytosis is of two types, phagocytosis and pinocytosis. Phagocytosis literally means cell eating, and phagocytosis is usually involved in immune responses. You will go through phagocytosis extensively in immunity, in the chapter of immunity, but we are discussing it briefly over here as well, that phagocytosis means cell eating. It involves the engulfing of solid particles. Okay, while pinocytosis is a cell drinking process, and it involves the uptake of liquid, while this involves the uptake of solid particles. And then the cell membrane extends around the particle and forms a vesicle called the phagosome. This is important, which is then brought into the cell. And pinocytosis, it is the uptake of liquid, and the cell membrane invaginates to form small vesicle containing extracellular fluid. So you need to remember that in bulk transport, vesicles are moved inside the cell or outside the cell. Now moving towards exocytosis, which is the other type of bulk transport. Exo means outside. So this is the process by which cells expel the materials in form of vesicles again, because bulk transport will always involve vesicles. Okay, so those vesicles may contain substances, and they fuse with the cell membrane. They release their contents to outside of the environment, and this is how exocytosis takes place. But this is not it. You need to remember these steps for endocytosis and for exocytosis. For endocytosis, the cell membrane engulfs the material from the external environment and forms a vesicle. Okay, for example, if this is the material, it's in the extracellular part. Okay, when it approaches the cell, all of this material will become a vesicle. Okay, in this vesicle, the vesicle formation, this is the second step. The membrane pinches off to form an intracellular vesicle. I already made it, intracellular vesicle. Then vesicle fusion. The vesicles may fuse with lysosomes. We discussed lysosomes, right, in chapter 1. So when these vesicles, these intracellular vesicles, they'll fuse with lysosomes or other organelles for processing or degradation. If, for example, this material is a waste material, if, for example, this is a waste material of proteins, then this intracellular vesicle will combine with lysosomes, and proteases will act upon this vesicle and will degrade it. Or sometimes an important thing might be coming into the cell, so it may fuse with some other organelle for processing. Okay, if degradation is involved, then lysosomes are going to play their part. Now moving towards exocytosis. For exocytosis, first step is vesicle formation. Vesicles containing these substances are formed inside the cell because in exocytosis, we are removing the material from the cell towards the extracellular part. Then vesicle transport happens, in which the vesicles move to the cell membrane along the cytoskeletal pathways, and then that vesicle is going to fuse with the cell membrane, and eventually all of the material is released outside the cell. Okay, so this is it. And if you have any questions, then just simply drop me the questions in the group or personally to me. Okay, I'll answer all of your questions if you have any query. But I believe that this was an easy chapter, and it's easy to understand, it's easy to comprehend. So till then, I'll call it a day. And till we have our next class, take care and Jazakallah khair.